You know how it is you're trying to stay informed, cut through the noise, and really understand what's happening in fields that matter.
Yeah, it's a constant challenge.
Today, we're tackling a big one, amyotrophic lateral sclerosis, or ALS. It's a brutal disease where the body's connection to its muscles progressively breaks down, leading to weakness, wasting, and tragically, typically within 5 years, the inability to breathe independently.
It's devastating. And we have some treatments, but
Nothing that truly changes the course of this illness, right?
Yeah.
So the search for effective therapies is absolutely critical.
it is. For you following along, understanding the devastating impact of ALS on individuals and their families really underscores why exploring any potential new treatment is so vital. A real breakthrough is desperately needed.
And that's precisely why we're taking this deep dive.
Mhm.
We're focusing on a recent phase 2 clinical trial investigating a novel cell-based product called CL-2020. Now, this isn't a conventional drug. It comes from some fascinating cells known as multilineage-differentiating stress-enduring cells. Thankfully, they're usually called Muse cells.
Much easier, yeah. New cells.
You shared an article detailing this trial, and we're going to unpack what its findings could mean for the future of ALS.
Exactly. The source you provided is a peer-reviewed original research article from the journal Cell Transplantation, published in 2023.
Right.
It lays out the details of this phase 2 trial, the safety measures put in place, and the initial clinical effects observed in a small group of individuals living with ALS. It gives us a valuable early look at this particular research direction.
Okay, let's unpack this. What are the key areas we're going to be exploring in this deep dive?
Well, we'll start by making sure we have a shared understanding of ALS itself, the different forms it takes and the current treatment options available.
Good idea.
Then we'll delve into the intriguing science behind Muse cells and how CL-2020 is developed. We'll examine the preclinical work that suggested this could be a promising approach.
The animal studies and stuff.
Exactly.
Yeah.
And of course, we'll thoroughly analyze the phase 2 clinical trial, its design, the people who participated, and what the researchers were specifically looking for.
Right.
Most importantly, we'll focus on the safety findings and the initial observations regarding any potential clinical impact and changes in relevant biological markers.
So what do they actually see?
Yeah. Our aim is to really understand the potential significance of these early results for the treatment of ALS.
Sounds like a plan. So before we get into this new research, let's just quickly revisit the fundamentals of ALS.
Right. As you mentioned, ALS is marked by the progressive degeneration of motor neurons, the nerve cells that control our muscles.
Okay.
It typically arises in middle age or later, and it triggers a series of increasingly severe issues, starting with muscle weakness and wasting, often in the limbs. This then progresses to difficulties with speech that's called dysarthria and swallowing, or dysphagia. Ultimately, the paralysis extends to the muscles responsible for breathing, which tragically leads to respiratory failure, usually within about 5 years of diagnosis.
5 years. Wow.
What's fascinating here is that we recognize two main categories of ALS. Sporadic ALS accounts for the vast majority, around 90% of cases, and in most of these, the cause remains unknown.
So it happens.
Pretty much, yeah. Then there's familial ALS, which makes up about 10% of cases, and these are linked to specific genetic mutations. We've identified a number of these genes, including SOD1, TDP43, and C9orf72, which are frequently involved.
Okay. And in terms of current treatments, we touched on this, but it bears repeating. While there are some options, nothing really halts the disease progression.
That's a crucial point to emphasize. We currently have a few FDA-approved medications, such as Riluzole, Edaravone, and the more recent combination of sodium phenylbutyrate and torosodiol.
Right, those names come up.
These can offer modest benefits, perhaps slightly slowing the disease's advance or helping to manage some symptoms, but they don't stop ALS, and their effectiveness varies from person to person.
So still a huge unmet need.
Exactly. This limited impact is precisely why the pursuit of new and more effective therapeutic strategies is so critical.
Okay, so that gives us the current picture. Now let's delve into this novel approach using Muse cells. What exactly are they, and why are they generating interest in the context of ALS?
Muse cells, which again stands for multilineage-differentiating stress-enduring cells, are quite remarkable. They were first identified in 2010 as a type of pluripotent stem cell found within mesenchymal stem cells derived from bone marrow.
Okay, so stem cells from bone marrow, but a specific kind.
Yeah, a specific subpopulation. What's particularly interesting is their unique molecular fingerprint. They express both SAA3, a marker typically found on human pluripotent stem cells. These are the master cells that can become any cell type in the body.
The really versatile ones.
Exactly. And they also express CD105, a marker associated with mesenchymal stem cells, which are known for their role in supporting tissue repair.
So they have traits of both.
Right. This combination is key because it suggests these Muse cells retain the master cell ability to become many different types of cells while also having some of the helpful characteristics of mesenchymal stem cells.
Interesting.
This dual expression gives them a really interesting characteristic. They have the potential to differentiate into cells representing all three primary germ layers. That's the endoderm, mesoderm, and ectoderm, meaning they have a broad developmental potential.
Okay, so they're like a versatile repair crew for the body. How does that translate into potential for treating a disease like ALS?
Precisely. Preclinical studies have demonstrated that when Muse cells are administered intravenously, they have this remarkable ability to seek out damaged tissues.
They actually find the damage.
Seems so. Once they arrive at these sites, they can then differentiate into the specific types of cells needed for repair and functional recovery. This has been observed in animal models of various conditions, including liver damage, hepatitis, muscle degeneration, and even heart attack, myocardial infarction.
So a track record in other areas.
Yeah. So the underlying idea with CL2020 is to harness this inherent therapeutic potential of Muse cells. CL2020 itself is a product that is specifically enriched with these SSCA3 positive Muse cells derived from bone marrow mesenchymal stem cells.
Got it. Enriched for the good stuff.
Yeah.
That sounds incredibly promising on its own, but what kind of evidence did they have that specifically pointed towards a potential benefit in ALS before they even began testing this in humans?
That's a very important question. The researchers had some compelling findings from studies using animal models of ALS.
The mouse models.
Right. They focused on both SOD1 transgenic mice, which are a common model for the familial form of ALS, and TDP43 transgenic mice, another significant model that reflects a different genetic pathway involved in the disease.
Okay, covering different types.
Exactly. In both of these models, they observed that intravenous injections of human Muse cells led to the cells successfully migrating to the spinal cord, specifically to the pia mater and underneath the white matter, which are key areas affected in ALS.
So they got to the right place.
They did. Even more encouragingly, repeated intravenous administration of these Muse cells in the SOD1 mice resulted in a significant improvement in hind limb muscle weakness.
Actual functional improvement.
Yes. And the article also mentions unpublished data showing that a single intravenous dose of CL2020 in TDP43 mice also significantly improved limb muscle weakness.
Okay, similar results in two different models.
Yeah. So the preclinical data seems to provide a strong rationale for exploring this in humans, with the cells showing both the ability to target the affected area and demonstrating a functional benefit in these models.
Wow, the fact that they saw functional improvements in multiple animal models is a pretty strong indicator. What else did they learn from these preclinical studies?
Well, they also reported on some of the potential ways these cells might be working. Muse cells appear to possess anti-inflammatory properties, and they can also help regulate immune responses.
So not just replacing cells, but maybe calming things down too.
Potentially, yes. Given that we increasingly understand that inflammation and immune system dysfunction play a significant role in the pathology of ALS, this adds another layer of potential benefit beyond simply trying to replace damaged motor neurons.
Makes sense.
This encouraging preclinical picture really set the stage and provided the justification for moving forward with human trials to see if these effects could be safely replicated in people living with ALS.
Okay, so the groundwork was laid. Now we get to the phase 2 clinical trial with CL2020 in actual ALS patients. Can you walk us through how this trial was structured and who the participants were?
Certainly. This was a single-center, open-label phase 2 trial conducted in Japan.
Open label, so everyone knew they were getting the treatment.
Exactly. Both the researchers and the individuals participating knew who was receiving the CL2020 treatment. They enrolled 5 individuals who had been diagnosed with ALS.
Just 5. That seems small.
It is very small, yeah. That's a key limitation we'll come back to. But each participant received intravenous infusions of CL2020 at a dose of 15 million cells once a month for a total of 6 months.
Okay, monthly infusions for half a year.
Right. The primary goal for the researchers was to assess the safety and tolerability of these repeated doses over a 12-month period.
Safety first. Makes sense for phase 2.
Absolutely. The key secondary goal was to examine the rate of change in the revised Amyotrophic Lateral Sclerosis Functional Rating Scale, or ALS-FRSR score.
Ah, the ALS-FRSR score.
It is. It's really critical in ALS research as it quantifies a patient's functional abilities across various daily living activities, things like speaking, swallowing, walking, dressing.
And they weren't just looking at that main functional score, were they?
No, absolutely not. They had a range of other secondary and exploratory endpoints. These included looking at changes in the ALS-FRSR scores for specific functions, such as bulbar function, speech and swallowing, limb movement, and respiratory function.
Breaking it down.
Yeah. They also measured percent forced vital capacity, percent VC, which is a key indicator of respiratory health, as well as manual muscle testing, MMT scores to assess muscle strength, grip, and pinch strength, and the modified Norris scale score, which is another way to track disease progression.
Lots of different ways to measure function.
And they even included a quality of life assessment using the 40-item ALS assessment questionnaire, or ALS-IQ-40.
That's a pretty comprehensive set of measurements.
What about biomarkers? Were they tracking anything in the patient's blood or spinal fluid to see if the treatment was having any biological effects?
Yes, this is where it gets really interesting. As exploratory endpoints, they collected blood samples at various time points to measure levels of certain cytokines, specifically tumor necrosis factor alpha, TNSA, and interleukin-6, IL-6.
The inflammation markers. Exactly, which are involved in inflammation, as well as sphingosine-1 phosphate, S1P, a signaling molecule that has been implicated in ALS.
S1P, okay, haven't heard as much about that one.
It's emerging as potentially important. They also obtained cerebrospinal fluid samples at the beginning of the trial and at several points during the treatment period to look at levels of ketotriacidase 1, CHIT1, and neurofilament light chain, NFL.
NFL, that's a big one for nerve damage, right?
It is, yeah. NFL is a marker often elevated when nerve cells are damaged, and CHIT1 is another potential indicator of neuroinflammation. So they were really trying to get a multifaceted understanding of what was happening biologically.
So they were aiming for a really detailed picture. What about who was actually eligible to be in this trial? Were there specific criteria that patients had to meet?
Yes, there were specific inclusion criteria in place to ensure a relatively similar group of participants as much as possible with just 5 people.
Right.
These included individuals aged 20 to 80 who had a diagnosis of probable or definite ALS based on established diagnostic criteria, a certain level of disease severity, symptom onset within the previous 24 months, and a baseline percent FVC of 80% or higher.
So relatively early stage, decent lung function to start.
Generally, yes, indicating relatively preserved lung function at the start of the study. Interestingly, they also had a criterion based on the rate of decline in their ALS-FRSR score. During a 3-month screening period, it had to be between -1 and -4 points, suggesting a certain level of ongoing disease progression.
Okay, so they weren't completely stable, but maybe not progressing super rapidly either.
Something like that, trying to get a specific window. Participants were allowed to be on a stable dose of rituzole, one of the existing ALS medications.
Ah, okay, so they could stay on that.
Yes. However, they were excluded if they had other inflammatory disorders, a recent history of cancer, active infections, or if they had previously received a bone marrow transplant or other stem cell therapy.
Okay, that makes sense. Trying to minimize other factors that could influence the results. Yeah. So with the trial designed and the participants enrolled, what were the key findings? Starting with how safe the treatment appeared to be.
This is a very encouraging aspect of the study. The researchers reported that the CL-2020 treatment was generally well tolerated.
Yes.
And they did not observe any serious side effects that were directly linked to the cell therapy itself. Over the 12-month follow-up period, there were a total of 28 adverse events reported across the 5 patients.
Okay, what kind of things?
The most common were headache, which occurred in 4 instances, and fatigue, reported in 3.
Pretty mild, generally.
Yes. There was 1 serious adverse event, a bone fracture in 1 patient, but after careful investigation, the researchers concluded that this was not related to the CL-2020 treatment.
Unrelated, okay.
Importantly, they did not see any significant changes in the patient's vital signs, medical examination results, electrocardiograms, oxygen saturation levels, or routine laboratory tests, including blood counts and biochemical markers.
So overall, the safety looked pretty good.
Yeah, from a safety standpoint, the repeated intravenous administration of CL-2020 appeared to be safe in this small group. This is a crucial first hurdle for any new therapy.
That's definitely a critical first step. What about the key secondary endpoint, the ALS-FRSR scores? Did they observe any changes in the rate of disease progression as measured by this scale?
Well, the ALSFRS-R scores, as is typical with ALS, did show a declining trend over the 12-month study period. That's expected.
Right, the disease progresses.
But when the researchers analyzed the rate at which these scores were changing, they observed a trend suggesting a potential slowing down of this decline at 12 months after the start of treatment, compared to the 3 months before the patients began receiving CL-2020.
A slowing trend. Okay, tell me more.
While this difference didn't reach the level of statistical significance in this small study group, remember, only 5 patients.
Right, hard to get significance with N in 5.
Exactly, but it's certainly an interesting signal. Looking at the individual patient data, they found that in 3 out of the 5 participants, the rate of ALS-FRS-R score change actually decreased, meaning they declined slower.
3 out of 5 showed slower decline.
Yes. 1 showed an increase in the rate of decline, so faster progression, and 1 showed no change. So while it's not a definitive result for the whole group, the fact that we're seeing a slower decline in some individuals offers a crucial glimmer of hope that this approach might be modifying the disease course.
So while not statistically significant for the entire group, there were definitely some individuals who seemed to be progressing less rapidly. What about those other clinical measures they were tracking, like respiratory function and muscle strength?
They observed a gradual downward trend in perf and total MMT scores over the 12 months, which, again, is generally expected in ALS.
Still declining overall.
Yes. Interestingly, in 2 of the patients, the perf appeared to be relatively stable, while it declined in the other 3. The total MMT score decreased in 4 of the 5 patients and remained unchanged in 1.
Some variability there, too.
Definitely. The total modified Norris scale score also showed a decrease over time, indicating disease progression. As for quality of life, as measured by the ALSAQ-40, the score showed a temporary decrease, suggesting a better quality of life at 1 month after starting treatment.
A little bump initially.
Yeah, but then it increased over time, indicating a subsequent decline in quality of life. This suggests a potentially complex and perhaps temporary impact on how patients perceived their well-being.
Interesting pattern. That's a lot of different data points. What about those biomarkers they were analyzing in the blood and spinal fluid? Did they see any significant shifts there?
Yes, there were some notable changes in the biomarker levels. In the serum, they found significant increases in both IL-6 and TNFY levels up to 6 months after the CL-2020 infusions.
Wait, increases in inflammation markers? That seems counterintuitive.
It does, yeah. This might seem a bit surprising given the reported anti-inflammatory effects of Muse cells in animal studies. It's something that needs more investigation.
Okay. What else?
However, they also observed a significant and continuous decrease in serum S1P levels over the entire 12-month period.
The S1P decreased continuously.
Yes. In the cerebrospinal fluid, they saw significant increases in CHAT1 levels at 4 and 6 months.
Another inflammation activation marker increasing.
Yes. While the levels of NFL, the nerve damage marker, showed a trend to increase, although this increase did not reach statistical significance.
Okay, so a bit of a mixed bag on the biomarkers. S1P down, but some inflammation markers up.
Kinda, yeah. It points towards complex biological effects.
Okay, so we have these changes in clinical outcomes, maybe a slower decline in some and in various biomarkers. How did the researchers themselves interpret these findings as a whole?
The researchers concluded that CL-2020 appears to be safe and that it may have the potential to slow down the progression of ALS, although it's unlikely to be a standalone cure or completely halt the disease on its own.
Realistic expectations.
Yes. They proposed that CL-2020 might be influencing some, but not all, of the underlying pathological processes involved in ALS. The sustained reduction in serum S1P levels was of particular interest to them.
Right, that continuous decrease.
They noted that CL-2020 expresses the S1P receptor 2, which allows it to strongly interact with S1P. They also pointed to a previous study where blocking S1P signaling showed beneficial effects in an ALS mouse model by modulating the immune response.
So the Muse cells might be soaking up or neutralizing the S1P.
That's their hypothesis, yeah. That administering CL-2020 might be effectively neutralizing S1P, thereby potentially dampening down S1P-related signaling in ALS patients.
Interesting mechanism.
They also highlighted the observation that the baseline levels of CHIT1 and NFL in the participants of this trial were relatively lower than what has been reported in natural history studies of ALS. Meaning that this particular group might have had a somewhat slower rate of disease progression to begin with, even before the treatment.
Okay, so maybe they selected for slower progressors, which could influence the results.
It's a possibility they acknowledged, yeah.
That's a really interesting potential mechanism involving S1P. But considering the small number of patients involved and some of the other aspects of the trial design, what were the main limitations that the researchers themselves acknowledged?
They were very clear about the limitations of this early stage study. The most significant was the very small number of patients who participated, just 5.
Yeah, we keep coming back to that.
It severely limits the statistical power of the findings and makes it challenging to draw firm conclusions about the treatment's effectiveness. They also pointed out the relatively short follow-up period of 12 months, especially for a disease like ALS that progresses over years.
Need to see longer term.
Definitely. The fact that it was an open-label trial, meaning there was no control group receiving a placebo, means we can't rule out the possibility of placebo effects or the natural variability in ALS progression influencing some of the observed trends.
The lack of a control group is huge.
It is. They also acknowledge that some of their analyses, particularly when looking at the rate of change in ALS FRSR scores, were conducted after the data had been collected, which is called post hoc analysis, and that can introduce potential biases.
Okay, so several big caveats. Small size, no control, short follow-up.
Exactly. Standard limitations for an early phase trial like this.
So based on these findings and the acknowledged limitations, what do the researchers suggest as the next steps in investigating CL-2020 as a potential treatment for ALS?
They strongly emphasize the critical need for a larger, double-blind, placebo-controlled clinical trial with a longer follow-up duration to truly determine whether CL-2020 provides a real therapeutic benefit for ALS.
The gold standard trial.
Pretty much, yeah. This type of study is the gold standard in clinical research and would provide much more robust evidence. They also suggested exploring the potential for combination therapies.
Using CL-2020 with existing drugs.
Exactly, where CL-2020 could be used in conjunction with existing ALS drugs such as edaravone or the sodium phenylbutyratotor-sodiol combination. The rationale here is that by targeting multiple disease pathways simultaneously, they might be able to achieve a more significant impact on the progression of ALS.
Attack it from multiple angles. Makes sense for a complex disease.
That seems to be the thinking, yeah.
Okay, we've taken a really thorough look at this phase 2 trial. Let's synthesize the key takeaways for you, our listener.
Okay. The main points to remember are these. First, CL-2020, this treatment derived from Muse cells, demonstrated a favorable safety profile in this initial phase 2 trial involving repeated intravenous administrations.
Safety looked good. Check.
That's a really important first step in evaluating any new therapeutic approach. Second, the study showed a preliminary indication, a hint, maybe that CL-2020 might have the potential to slow down the progression of ALS in some individuals.
Some signal on slowing progression, but not definitive.
Right. And this potential effect might be linked to the modulation of S1P signaling, which is an intriguing area for further research.
The S1P connection is interesting.
However, it's absolutely essential to keep in mind that these findings are not conclusive due to the study's limitations, most notably the small number of participants and the absence of a control group.
Huge caveats.
Definitely. More rigorous investigation through larger controlled clinical trials is definitely needed to confirm whether CL-2020 truly offers a meaningful therapeutic benefit for people living with ALS.
Right. So for you, the learner, this provides a really interesting update on a therapeutic strategy that's still in the early stages of development for a truly devastating disease.
Yeah, very early stage.
The aha moment, for me, is grasping the unique potential of these Muse cells and how they might be interacting with pathways like S1P signaling in the context of ALS. It really underscores the ongoing innovation in the quest to address this complex condition.
Precisely. And this leads us to a final thought for you to consider. Given the multifaceted nature of neurodegenerative diseases like ALS, with potentially numerous underlying mechanisms contributing to their progression.
Yeah, it's not just one thing going wrong.
Exactly. How crucial do you think the development of combination therapies, those that target multiple of these pathways concurrently, as the researchers suggested, might be in ultimately making a substantial difference in the lives of individuals affected by this disease?
Combining forces, basically.
Yeah, it certainly gives us something to think about as we continue to follow the advancements in stem cell therapies and the broader landscape of ALS research.